Hello students! Today we're going to look at something really cool. We're going to look at metabolism. So metabolism is actually because we're going to look at two parts of it. First of all, you need to know that every living cell is like a tiny little chemical factory where thousands upon thousands of chemical reactions are occurring every second. The cell is always taking energy that is stored out in sugars and takes that energy to perform work. And metabolism is an emergent property of life that arises from the orderly interactions between molecules. And what is metabolism exactly? So, for instance, we have a pathway of metabolism. We'll start with a molecule. We'll dump an enzyme on it. We get a reaction. We get another product, B. And then we put another enzyme, another reaction. We get another product, C. And then we put another enzyme, another reaction. We get another product, D. And that just is a pathway. So a pathway, a metabolic pathway, starts with something and ends with something, and in between you get all these steps where there's speeding up by specific enzymes. So metabolism is actually composed of two parts, anabolism and catabolism. Catabolic pathways release energy by breaking down complex molecules into simpler compounds, as they do in cellular respiration, where the breakdown of glucose occurs in the presence of oxygen, and we get some energy that is released at that time. Anabolic pathways consume energy. And why do they do that? They need to build complex molecules from simpler ones. So they're actually doing some work. And they, for instance, you could string a bunch of amino acids together and you could synthesize a protein. So something big from something small. Bioenergetics is the study of how energy flows through the living systems. But first of all, what exactly is energy? We've always talked about energy, but we don't quite know what do we mean by energy. Energy is defined as the capacity to cause change. It exists in various forms, some of which can perform work, and the energy can always be converted from one form to another. But usually, generally speaking, energy is either potential energy or it's kinetic energy. So there's two big classes of it. Big, broad categories, kinetic and potential. In the kinetic category, we have the energy of motion, the energy that's radiant, energy thermal, and the energy of sound. So these are all things that are moving. Potential energy is the energy that is residing inside chemicals, or is a mechanical energy where you push or pull something. So that's potential energy. It has the potential to do work. Gravitational energy is also a kind of potential energy. Nuclear energy is a kind of potential energy, and electrical energy is also potential. Kinetic energy is associated with motion. Heat, which is also called thermal energy, is energy associated with random movement of atoms or molecules. Potential energy is energy that the matter possesses because of its location or its position. Chemical energy is potential energy because it is available for release in a chemical reaction. Here are some pictures of commonly seen forms of energy. So the first one, you see a magnet and some pilings. The next one, the middle one, you see a bullet actually cutting through a card from a deck of cards, and that's kinetic, so it's moving. Heat, you see a match that's lit up. Actually, there's light in there too, but it's also producing heat. But both are kinetic energy. Light in the bottom left-hand corner, sunshine coming through these trees, that's a form of kinetic energy. The skydivers that are jumping, hurtling towards the earth are exhibiting gravitational or potential energy. And chemical energy is stored inside the vegetables that we're going to eat, so that is a form of potential energy. Other forms, sound. the guitar, the ukulele that you see. It's a form of kinetic energy, electrical energy from the bumping of two clouds that are charged in opposite ways. The elastic potential energy from pulling an elastic, nuclear energy, wind, geothermal. There's all kinds of energy that we can look at. Energy is all around us. In this picture, you can see energy changing from one form to another. So at the very bottom of the picture, you see somebody climbing up a ladder out of the water. And so this person is converting kinetic energy of movement into potential energy because she's getting higher and higher up on the platform. The guy that's all the way on the diving board has more potential energy standing on the platform than he would if he stood at the second platform, or even if he was on the water. And the diver, you see, is converting potential energy into kinetic energy. The little heads that you see bobbing in the water, they have less potential energy in the water than they would on the platform, because potential energy is energy of location. And this brings us to the laws of thermodynamics. Thermodynamics is the study of energy transformations. In an isolated system, such as like a liquid in a thermos, that is unable to exchange energy or matter with its surroundings. So that's called an isolated system. Technically, there are no real isolated systems, but a thermos would be an example of an isolated system for a short amount of time. But an open system, energy and matter can be transferred between this system and its surroundings, such as organisms. We're open systems, so we're always dropping matter off, shedding skin, releasing heat, taking energy in the form of food. So we're just open systems. The first law of thermodynamics is very important because what it says is that the energy of the universe is constant. And so therefore, energy cannot be created, it cannot be destroyed, and it can only change form. It's also called the law of conservation of energy. Here you see a depiction of it. You see a big old bear. He's chewing on some fish, so he's going to be releasing the chemical energy that's stored in the fish into work, or kinetic or swimming energy, or some sort of energy that he's going to use for metabolism. So that's the first law of thermodynamics in action. The second law of thermodynamics is very simple. All it does is it takes the first law and it extends it. During every energy transformation, some energy is lost as heat, and it's never able to be used again. So in every energy transfer or transformation, what you will get is an increase in entropy of the universe. So the entropy of the universe is going to keep on increasing. So here, what you see is the same bear, and he is eating his fish. And now he's transforming that chemical energy, which was potential, into metabolic energy. So he's keeping his body temperature constant, which has been given off as heat. And he's breathing in oxygen, breathing out carbon dioxide and water. So here we are. We're looking at the second law of thermodynamics, where heat from his body is just lost to the surroundings, and he can never get that back. So that's just adding to the entropy of the universe. There is just no way to avoid loss of usable energy and to avoid increasing entropy of the universe, because it just works that way, that the universe is moving towards something, well, theoretically towards heat death, where all the energy will have been converted into energy that cannot be used for work anymore. Spontaneous processes would be those that can occur without energy input. And so they can happen either quickly or slowly. But for any process to occur without energy inputs, it has to increase the entropy of the universe. So there are actually 3 laws of thermodynamics, and here are all of them. We won't actually be using the third law. It's just here for completeness. The first law of thermodynamics, it just means energy cannot be created nor destroyed. It can only change form. And it is also known as the law of conservation of energy. The second law of thermodynamics just simply states entropy of a system will always increase at every transformation of energy. And the third law of thermodynamics says entropy will approach zero as the absolute temperature approaches zero. So the reason we don't really concern ourselves too much about the third law of thermodynamics is because the absolute temperature is never going to be zero anywhere. There are some icy bodies in interstellar space that are close to zero, but not zero, not absolute zero. So if you can't get to 0 degrees Kelvin, what that means is there's always some motion, random motion of the molecules going on. And that means that entropy is still never going to be zero, even at that point. So we won't concern ourselves with the third law because we mostly use the first and the second law of thermodynamics in daily life. Cells create ordered structures from less ordered materials, and that is called anabolism. They also replace ordered forms of matter and energy with less ordered forms, and that's called catabolism. The catabolic pathway in a cell releases free energy in a series of reactions. Energy flows into an ecosystem in the form of light and exits the ecosystem in the form of heat. So there's this other concept of free energy, or Gibbs free energy. A living system has some energy that can do work when the temperature and pressure are constant. But the change in free energy in any process is related to the total energy of its system, its entropy, and its temperature, and it's given by the mathematical formula that is depicted on the third bullet. Delta G, which is the change in Gibbs free energy, is equal to delta H, which is the change in total energy, minus T, which is temperature in Kelvin, times delta S, which is the change in entropy. So that is just putting in mathematical words, depictions, symbols, what I just said in words. Gibbs free energy is a measure of chemical energy. All chemical systems, that would be all life, tend naturally towards states of minimum Gibbs free energy. We like to go to minimum Gibbs free energy states because that is where stability is. So here is another way of looking at, if we don't put the deltas in, it's the same formula as we saw in the previous slide. So no deltas, but its Gibbs free energy is equal to the enthalpy, or the heat content, minus the temperature times the entropy, or you could think of entropy as randomness. So the change in free energy, or the delta G, or Gibbs free energy, the change in enthalpy accounts for energy transferred to the environment through either expansion or heating at constant pressure. Enthalpy is a term. It's a thermodynamic potential describing energy transfer, given by this formula, which you can see at this place called, I'm sorry, it's H. So enthalpy is H, or the thermodynamic potential, which is equal to U, which is the internal energy of that system, time plus pressure times the volume. So that will actually give you what the enthalpy is. Free energy is the measure of the instability of a system, its tendency to change to a more stable state. So during a spontaneous change, free energy decreases and the stability of the system increases. Equilibrium is a state of maximum stability. A process is spontaneous and can perform work only when it is moving towards equilibrium. Some reactions occur spontaneously. Others require an input of energy. Only the processes with a negative Gibbs free energy change are spontaneous. And here is what I just said in words in a graph form. So if you look at the x-axis, it's just telling you the reaction, the state of the reaction, so how far has the reaction proceeded. And on the y-axis, you see the Gibbs free energy. So what you see is a reaction starts at 0, and the Gibbs free energy is decreasing and decreasing until finally it comes to this well, which is at the bottom of a well, and that is where equilibrium is. Here Gibbs free energy is 0, and we like to stay in a stable state. However, on the other arm, on the right hand side, you see Gibbs free energy is greater than 0, and at that point it will also tend to move towards the well where Gibbs free energy is 0 and to equilibrium. So to summarize, more free energy, or the higher Gibbs free energy, the less stable it is, the more work capacity can do. In a spontaneous change, the free energy of the system decreases, the system becomes more stable, and the released free energy is used to do work. And if there's less free energy, or there's a lower Gibbs free energy change, the system is more stable, but it has less capacity to do work. So here is another illustration. On the left-hand side, you see this, it's just a closed system, and you see two cylinders. One is full of water, the other one is not, and in between there is a third cylinder with a turbine or some sort of device that is moving. And the second cylinder is at a lower level than the first. So since the first cylinder is full of water and the opening of the turbine is below the top of the water level, the wheel will move and the water will move into the empty cylinder until equilibrium is reached and the water level is the same in both cylinders. Then no more work will be done. So because equilibrium has been reached, there is no more work that can be done, and Gibbs free energy is therefore equal to 0, whereas before Gibbs free energy was negative, it was less than 0, and it could do work. These are also known as exergonic and endergonic reactions. So an exergonic reaction proceeds with a net release of free energy, and it is spontaneous. An endergonic reaction absorbs free energy from its surroundings, and it is not spontaneous. So usually they're harnessed together inside a cell. But here we have a picture, a graph of an exergonic reaction. First on top, where energy is released, and this reaction is spontaneous. So you see a big pink orangish bar of free energy in the reactants, and the reaction starts to progress. Energy is released and you get products, and you see that a whole bunch of energy is released, so Gibbs free energy is less than 0, or it's negative. So that means it's exergonic and spontaneous. In the bottom graph, which is blue, what you see is a small bar to begin with, and those are the reactants, and you have to get products. Well, you're not going to get products unless you add energy to it, because the amount of energy you'll need is more than 0, so it is non-spontaneous, and the reaction is therefore endergonic. Another depiction of the same graph, but in a different way. So on the left-hand side, which you see the same idea, we have reactants that are going to form products, and Gibbs free energy is less than 0, and so therefore the product will form and the reaction is spontaneous and it is exergonic. But on the right hand side, what you see is a funny looking graph where the reactants are all the way down below the dashed line, and there's a tremendous amount of activation energy needed for the reaction to start, and then products can be formed. So Gibbs free energy is greater than 0, so the reaction cannot be spontaneous, and it needs an influx of energy to get it going. Cells are never in equilibrium. Life is always in disequilibrium because cells are open systems with a constant flow of materials. And a defining feature of life is that metabolism is never at equilibrium. Entropy will always win in the end, but for as long as we're alive, we can live temporarily in disequilibrium with the environment, and that is the property of life. A cell does 3 main kinds of work. It does either chemical work, or it transports stuff, or it does mechanical work. And to do work, cells need to manage energy resources by coupling the reactions. So they use an exergonic process to drive an endergonic process, which otherwise would never be able to happen. Most energy coupling in cells is mediated by ATP, the currency of life. ATP is adenosine triphosphate, as you remember, and it is the energy shuttle of the cell. It is made out of ribose, and we looked at this before, a hexose sugar, adenosine, adenine, which is a nitrogenous base, and 3 phosphate groups, which you see strung on the left-hand side. When ATP is hydrolyzed, it becomes ADP, and energy is released, and 1 inorganic phosphate is also released. So the bonds between the phosphate groups of the ATP tail is broken by hydrolysis, and energy is released from ATP when the terminal phosphate bond is broken. This release of energy comes from the chemical change to a state of lower free energy. So that's kind of cool. In the cell, the energy from the exergonic reaction of ATP hydrolysis is used to drive an endergonic reaction. So that is why ATP is called the currency of life, because every time you hydrolyze an ATP molecule, you're going to get some energy. And that energy is then, you can use it to go into disequilibrium reactions, reactions that would never occur otherwise. And here you have energy aplenty in good supply, and so you can go ahead and go into that reaction. ATP drives endergonic reactions by phosphorylation. It transfers a phosphate group to another molecule. The recipient molecule is now called a phosphorylated intermediate. For example, glutamine, which is stored in muscles and also in the lungs, is important for removing excess ammonia. And glutamic acid, which is another amino acid, is a neurotransmitter. And so here we have a picture. On the top you see glutamic acid binding with ammonia to form glutamine, and you can see that the Gibbs free energy is plus 3.4 kilocalories per mole. So glutamic acid being converted to glutamine, and the Gibbs free energy is plus 3.4 kilocalories per mole. But in the center, what you see is glutamic acid with ATP then becomes a phosphorylated intermediate and plus ADP, and then you add ammonia to it, and what you get is glutamine plus ADP plus phosphate. So when you couple this reaction with ATP hydrolysis, that's what happens. And at the very bottom, what you see is the first reaction, which was plus 3.4 kilocalories per mole, the second reaction, which is negative 7.3 kilocalories per mole, and if you add it all up, you get negative 3.9 kilocalories per mole, which is the free energy change for the coupled reaction. transport and mechanical work in the cell are also powered by ATP hydrolysis. ATP hydrolysis leads to a change in protein shape and its binding ability. So here we are with some pictures of that. And the first picture on the left-hand top side, you see a transport protein. You add some ATP to it. And what will happen is it will open up. Okay, so some work is done, it opens up, and the solute is transported. And of course, you end up with ADP and a phosphate group. In the bottom picture, what you see is something very, very cool, a motor protein working. What you see is an addition of ATP to a motor protein, and this motor protein is actually going to jump up on the cytoskeletal track one step, and it will change ATP into ADP, release one inorganic phosphate, and with that energy it can move up the track. So that is mechanical work where ATP is binding non-covalently to motor proteins, and then it is hydrolyzed. ATP is a renewable resource. It's regenerated all the time by the addition of the phosphate group to adenosine diphosphate. The energy to phosphorylate ADP comes from catabolic reactions in the cell. The ATP cycle is a revolving door through which energy passes during its transfer from catabolic to anabolic pathways. That's just another way of looking at it. And so here we have what I just said in words in a nice pretty picture. So on the left hand side you have this zigzaggy arrow which represents energy from catabolism, like exergonic energy releasing processes, forming ATP plus water, and then releasing energy with the hydrolysis of ATP to become ADP and phosphate, and then that energy is going to be used for endergonic processes. Enzymes will speed up the metabolic reactions by lowering energy barriers. What does that mean? An enzyme is a catalyst. It's nothing but a catalyst. A catalyst is something that speeds up a reaction without being consumed in the reaction. An enzyme is a living catalyst, so it's a protein. And for instance, the hydrolysis of sucrose by the enzyme sucrase is an example of an enzyme catalyzed reaction. There are hundreds and hundreds of them, but this is just a simple one. I have a picture of it. Here is a sucrose molecule. Notice the little O being held by the two molecules of monomers of sugar. You add some water to it, you add an enzyme sucrase, and all of a sudden they break down into two monomers, glucose and fructose. And this is what they originally came from. When you look at the reaction, this is a reaction that can happen because the substrate, notice what happens to the Gibbs free energy. Without the enzyme, the uncatalyzed reaction, you need a really a huge amount of energy transfer to activate this reaction. But with the enzyme, the catalyzed reaction doesn't require that much. And that is called the energy activation barrier. So every chemical reaction involves a bond that breaks and a new bond that's formed. The initial energy needed to start a chemical reaction is called the free energy of activation, or activation energy, and it's usually written as capital E sub capital A. Activation energy is often the thermal energy that the reactant molecules absorb from their surroundings, like this. So you have reactants A, B, and C, D, and it's an exergonic process. So you're going to start this reaction. You get a transition state, and then you get products. And instead of AB, you get AC, and instead of CD, you get BD. and there is an energy change. The Gibbs free energy is less than 0, so it's negative. So this reaction would actually occur because it's exergonic. Enzyme speed reactions, they catalyze them because they lower this barrier. They don't affect the change in the free energy. Instead, all they do is they hasten reactions that were going to occur anyway. And that's important to remember. They just lower the activation energy barrier. They do not change the Gibbs free energy at all. Okay, so here is it showing you the red line shows what happens with an enzyme. The black line shows what happens if you don't have an enzyme. A reaction will occur. It needs a ton of energy to work, and you get products. But with the enzyme, the reaction occurs, but you don't really need a lot of energy to start it. But notice, under the dashed line, that is what is Gibbs free energy. That is unaffected by the enzyme. It's still the same. You get the same number. The reactant that an enzyme acts on is called the substrate. The enzyme will bind to a substrate. Enzymes are very specific. They only bind to certain substrates. And when they do, they form an enzyme-substrate complex. And each enzyme catalyzes a very specific reaction. How does an enzyme do it? You would wonder. Well, enzymes have very small active sites, usually only 2 to 4 amino acids long, and they may be a really big protein molecule. But the active site is the region where the substrate will actually bind, and that is very small. What it does is, when the substrate binds to that active site, it will induce a fit of the entire protein. The substrate will make the enzyme fold a little bit differently into a transitional state, and that's because the chemical groups of the active site will actually move to do this. It's just more favorable to do it that way, like this. So the blue part with little balls is an enzyme, and you can see the active site, which looks like its mouth, and you see the little red stuff, which is called a substrate. Well, when the substrate actually goes and binds with the active site, notice what happens to the enzyme. It looks like it's going chomp. It folds over it and becomes a complex, and then it will, the bonds are actually doing this for them, the hydrogen bonds and the other weaker forces. And then it will break apart the bond that is in the substrate and release it. It's ready for the next molecule to show up. In an enzymatic reaction, the substrate binds to the active site of the enzyme. The active site lowers the energy of activation by orienting the substrates correctly, straining the substrate bonds, providing a favorable microenvironment, that would be ionic, and covalently bonding to the substrate temporarily. So, number 1, here we are. We have 2 substrates entering the active site. One is green, one is sort of a pinkish color. The enzyme is this purplish blue blob. And the 2 substrates will go and bind in the active site, and you get an enzyme-substrate complex. And the substrates are held there by weak interactions. And then they're converted into products while there. And then the products are released, and the enzyme is now available again for new substrates. And this goes on all the time. The activity of an enzyme can be affected by general environmental factors like temperature, pH, and chemicals. Optimal conditions favor the most active shape for that enzyme molecule. So the effects of temperature and pH, let's look at those. Each enzyme has an optimal temperature in which it can function. Usually, all human enzymes function best at 37 degrees centigrade. But that's for humans, not for every organism. Each enzyme also has an optimal pH in which it can function. Usually, enzymes function best between pH 6 and 8. However, pepsin, which is in the stomach, can only function at pH 2. Trypsin can only function at pH 8. But usually, all human enzymes function best between pH 6 and 8. And here is a graph showing exactly what I just said before. The top graph, it shows the optimal temperature for a typical human enzyme, 37 degrees. The red line actually shows you the optimal temperature for an enzyme of a thermophile, which is not 37 degrees, but actually 40 degrees above that, so it's 77 degrees. So it's a pretty hot place, but that's the optimal temperature for that extremophile. And then at the bottom, you see another graph, and what you see is the optimal pH. The black line shows you the pH for pepsin, which is 2, and the red graph shows you the pH for trypsin, which is 8. So most enzymes actually prefer 6 to 8, but every enzyme is very specific and very picky. There are also some other things that help enzymes, and they are called cofactors. Cofactors are non-protein enzyme helpers. They can be inorganic, like metals, or they can be organic. But if they're organic, then they're called a coenzyme, or coenzymes or vitamins. Vitamins are part of coenzymes. Because there is the active site, which is small in the enzyme, there are other things that can also compete to go bind to that active site. They may have a similar shape, but not be quite the best fit, but they are going to interfere with the active site really binding with the real substrate. So that's called competition, and it's called competitive inhibiting, because they will try to bind to the active site, and they won't let the substrate bind to it. But non-competitive inhibitors bind to a different part of the enzyme, but that still doesn't help, because it causes the enzyme to change its shape, and then the active site becomes less effective. So those were examples, let's see, of inhibitors would be toxins, poisons, pesticides, antibiotics. So these are all inhibitors which will bind to the active site of an enzyme to compete with the substrate. In antibiotics, we would rather want that, because we don't want the parasitic pathogen protein binding with the enzyme. We'd rather have the antibiotic binding there, so we can get rid of the pathogen. Here is inhibition of an enzyme activity. So there's several kinds. The first is just normal binding. Okay, in the first picture on the left, you just see normal binding. You see an active site, you see a substrate, and you see an enzyme, and it's a perfect fit. But in competitive inhibition, what you see is in the substrate, which is still that turquoise-looking blob, is now competing for the same site with this big old blue circle-ish thing, which fits perfectly on one part of the active site, but not the entire. So it's not really a good fit, but it's a good fit somewhere. So that's competitive inhibition. Non-competitive inhibition, well, there'll be another molecule, but it's going to bind to not the active site, but somewhere else on that enzyme. It shouldn't, but it does. And so the entire shape of the enzyme changes, and now the substrate can't bind to it. As you can see, what happened to it. Now it doesn't even look like our good old enzyme in the beginning. So enzymes are proteins that are encoded by genes. And any changes in genes will lead to changes in the amino acid composition of the enzyme. And any alterations in the amino acids will result in either new enzyme activity or completely altered substrate specificity. Under the new environmental conditions, the new enzyme might be favored, but it might not, in which case it'll just be gotten rid of. Why do we have a control of metabolism? We need that, because if we didn't have that, we would have chemical chaos. There's so many chemical reactions going on inside a cell, so much metabolism going on. If it's not tightly regulated, you would have a bunch of chaos. Cells will switch the genes on or off that encode for specific enzymes which regulate the activity of the enzymes. They also use chemical regulation and mechanical regulation. So chemical regulation of enzymes, what does that mean? And that means that when a regulatory molecule binds to a protein on one site, it affects the function of the active site of a protein, and that is called allosteric regulation. So what's happening is it's a regulatory molecule, and it's going to bind to a protein at one site, but it's going to affect the function of the active. It won't bind to the active site. It'll bind to some other site, and it will regulate the enzyme itself. So that's called allosteric regulation. Allo means somewhere else. Allosteric regulation can inhibit or stimulate the activity of an enzyme. Each enzyme has an active and an inactive form. The binding of an activator stabilizes the active form. The binding of an inhibitor stabilizes the inactive form of the enzyme. Another form of allosteric regulation is called cooperativity, that amplifies the enzyme activity. Here, one substrate molecule primes an enzyme to act on additional substrate molecules more easily. So it's like cooperation. It's also allosteric because the binding by a substrate to one active site affects catalysis in a different active site. So this is also allosteric. Here is a picture of all those things I just talked about. So we see on the left-hand side allosteric activators and inhibitors. So let's look at them. You see this pretty little picture. It looks like a clover leaf with four leaves on it, but it's actually an allosteric enzyme with 4 subunits. And there's one active. There are actually 4 active sites, but one is active at the moment, and then there are regulatory sites, and there are 4 regulatory sites. And what you see is the activator coming. It's this little green and triangular shaped molecule, and it's going to wedge into the regulatory site, and that will make the enzyme into a stabilized form. However, you could also have an inactive form where the enzyme doesn't even look like our nice active enzyme. It has a non-functional active site, and it actually has an inhibitor, which will bind to the regulatory site, and it will stabilize the inactive form. And on the right-hand side, what you see is another form of allosteric regulation, cooperativity, where you see the inactive form of the enzyme, and you see a substrate binding to it, and you get a stabilized active form. Enzymes regulate their own production by negative feedback. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing more product than is needed. In feedback inhibition, the end product of a metabolic pathway shuts down that pathway. Let's see if I can get this thing to move. My goodness gracious, what happened? All right, here is a picture that shows you an enzyme. So the purplish looking blob is the enzyme, and it has an active site. That's at the top of the page. Well, it moves into this stabilized form where something is binding to its active state. And then it forms an intermediate complex, A, B, C, D, and in the end you get out of threonine, you get isoleucine, which is the end product. But the isoleucine is actually going to bind to the same enzyme in an allosteric site. So now the active site is no longer available because the pathway is halted. It's no longer nice and flat like a bed, but it's all curved up. And so the isoleucine is used up by the cell. And then once there's no more isoleucine, then that allosteric site will be empty. The enzyme will reconfigure into its regular shape, and threonine will bind to it, and you'll get more isoleucine again. So that's just how a pathway would work for this. Control by localization. What does that mean? That means the structures within the cell help bring order to metabolic pathways so they can segment reactions into specific areas. You don't want a bunch of chemical reactions happening hither and thither. You would rather have them just be confined to an organelle or inside a wall so that it doesn't affect other reactions or it doesn't get messy and tangled. Some enzymes act as structural components of membranes. In eukaryotic cells, some enzymes reside in specific organelles. For example, enzymes for cellular respiration are located in the mitochondria, as in here. And here we're going to look at this last slide where you see a mitochondria on the left-hand side, which is the orange-looking thing, and then blown up under an electron micrograph on the right-hand side. What you see is it's like the cigar-shaped organelle with a lot of walls inside, and the matrix has enzymes in solution that are involved in one stage of cellular respiration. And then the enzymes for another stage of cellular respiration are actually embedded inside those little thin lines, those walls of the inner membrane. So that is how order is brought to metabolism inside a cell. Goodbye, students.